Hey everyone, and welcome to today's Explainer. We are bringing absolute energetic enthusiasm to our breakdown of today's fascinating source material. Aaj hum ek an incredible journey per Nakan Levelihan. We're going to start from the absolute microscopic foundations of a single cell and build all the way up to the grand categorization of all life on Earth. We're going to break down some major biological myths and look at life through a completely new structural lens. Are you ready? Let's dive in. Let's kick things off with a pretty simple question. Kya ek murgi ka anda, a hard-shelled chicken egg, एक closed thermodynamic system hai? I mean, it looks perfectly sealed from the outside, right? Well, the answer is a resounding no, because a growing embryo inside actually needs oxygen and releases carbon dioxide, which is an exchange of matter, and it exchanges heat with its surroundings. That egg is actually an open thermodynamic system. And here is the big secret. Every single living cell on Earth acts in this exact same way. If a cell ever truly becomes a closed system, it literally ceases to be alive. All right, Section 1, the ultimate definition. We are stripping the cell down to its absolute basics. To really understand biology, we have to strip away all the complex, confusing parts. Broadly speaking, what actually is a cell? It's simply an aqueous compartment, basically a water-based environment, wrapped up tightly in a boundary, functioning as an open thermodynamic system that constantly trades energy and matter to survive. Notice we didn't mention a nucleus, or DNA, or mitochondria, or any of those organelles. This right here is the absolute bedrock definition of a cell. Now, there is a very strict rule regarding that boundary. We hear the term unit membrane thrown around a lot, but you can't just call any biological wall a unit membrane. To qualify, it absolutely must be composed of a lipid bilayer. Carbohydrates? Totally optional. Proteins? Optional too. But a boundary made purely of protein or just cholesterol, that simply does not qualify as a unit membrane. This actually helps clear up a really common misconception, especially when we think of plant or bacterial cells that have those tough, rigid outer cell walls. That cell wall? It's just armor. It's not the real boundary. The plasma membrane is the definitive border of life. If you were to magically strip away the plasma membrane and leave only the cell wall, the cell instantly loses its existence. Moving on to Section 2, the rule breakers. Let's talk about entities without a cellular body. So, not everything on Earth fits neatly into that beautiful little aqueous compartment we just defined. Enter the non-cellular entities. These are obligate intracellular parasites that have absolutely no true cellular body of their own. Let's contrast viruses with viroids. We all know viruses, right? They're nucleoproteins, meaning they have genetic material wrapped up safely in a protein coat, and they can infect almost anything, from humans to bacteria. But viroids? They are just tiny, naked, circular loops of RNA, no protein coat whatsoever. And fascinatingly enough, they exclusively plague plant cells. So just how tiny are these viroids? We're talking fewer than 500 nucleotides. To put that into perspective, they are the absolute smallest known infectious agents on Earth. They are essentially the tiniest organisms we know of existing entirely without a cellular body. It's mind-blowing. Section 3, the Great Divide and Whittaker's 5 Kingdoms. Let's organize the cellular world. Having covered those fascinating little exceptions, how do we begin to organize the millions of cellular life forms on our planet? Well, it starts with clearing up a major myth. The word prokaryote literally translates to before nucleus. People often mistakenly think prokaryotes have a primitive or incipient nucleus. They don't. They simply don't have one at all. Eukaryotes, on the other hand, are strictly defined by having a true nucleus that houses their genetic material. Building on this big divide, back in 1969, Robert Whittaker organized all cellular life into his famous 5 kingdoms: Monera, Pertista, Plantae, Fungi, and Animalia. He did this using 4 foundational pillars. He looked at whether they had a nucleus, how many cells they had, how they produced or found their food, and finally, how they actually took that food into their bodies. Like, are they fungi simply absorbing it, or animals actively ingesting it? Which brings us to Section 4, Redrawing the Map, Carl Woese and the 3-Domain System. Science never sits still, right? Fast forward to 1990, and a scientist named Carl Woese realized that Whittaker's kingdom Monera, which housed all the prokaryotes, was hiding a massive, revolutionary biological secret. Woese didn't look at the whole cell. Instead, he looked deep inside the 30S ribosomal subunit, specifically at something called the 16S ribosomal RNA. When he analyzed the sequence of this RNA, he found unique oligonucleotide signature sequences. This proved that the organisms we blindly grouped together as bacteria were actually two completely, fundamentally different domains of life: true bacteria and a newly discovered group called archaea. And this fundamentally rewrites the true evolutionary tree. We often picture a linear progression in our heads, like first came archaea, then they turned into bacteria, and then bacteria eventually turned into eukaryotes. Actually, scratch that. That is entirely wrong. From the last universal common ancestor, bacteria branched off on their own, while a separate common ancestor branched off and later split into archaea and eukaryotes. This means archaea are actually more closely related to us, eukaryotes, than they are to bacteria. So the absolutely crucial point here is that archaea live a fascinating dual existence. Structurally and metabolically, they look and act like bacteria. No nucleus, similar cell organization. But when it comes to their genetic machinery, how they actually replicate DNA and transcribe RNA, they are almost identical to eukaryotes. Because of this, an antibiotic like rifampicin, which destroys bacterial RNA polymerase, is completely useless against archaea. Why? Because archaea's genetic machinery looks like ours, not the bacteria's. Let's wrap things up with Section 5, the cellular diet. How cells actually survive and consume. Let's circle all the way back to thermodynamics. Every open system needs fuel. Biologists use a really strict matrix to categorize life based on 3 absolute requirements. First, where does a cell get its energy? If it's from light, it's a phototroph. If it's chemical, a chemotroph. Second, where does it get its carbon? Inorganic sources make it an autotroph. Organic sources make it a heterotroph. And third, where does it get its electrons? Inorganic means lithotroph. Organic means organotroph. Now let's challenge something you probably learned back in elementary school. An autotroph is not simply an organism that makes its own food. The strict, true definition is an organism that derives its carbon exclusively from inorganic sources, like pulling carbon right out of CO2 in the air, or literally extracting it from calcium carbonate rocks. And this completely shatters our standard assumptions. Like, are all plants autotrophs? No way. Look at parasitic plants like Cuscuta or the giant Rafflesia. They have no chlorophyll. They do not photosynthesize. Because they cannot utilize inorganic carbon from the air, they have to steal organic carbon from other living things. That makes these plants heterotrophs. So let's apply our new biological matrix to the real world. Cyanobacteria and green plants are photolithoautotrophs, meaning they use light, inorganic electron donors like water, and inorganic carbon. But look at humans and fungi. We both get our energy from chemical oxidation, our electrons from organic molecules, and our carbon from organic sources. In the grand thermodynamic matrix of life, you and a mushroom are the exact same thing: chemo-organo-heterotrophs. Which brings us to the end of our journey today. We've stripped life down to an aqueous compartment and built it all the way back up into a master matrix of energy and matter. So, अगली बार जब आप किसी पौधे या जीव को देखें, don't just see a species. सोचें as an open thermodynamic machine. Where exactly is it stealing its carbon and its energy? Keep observing, keep questioning, and I'll catch you next time.